So let's talk about a few questions of CS and NET syllabus, that is on RNA editing, splicing, capping and polyadenylation. So first question: the addition 3' end of most eukaryotic mRNA is defined by the addition of a poly A tail, a processing reaction called polyadenylation. The addition of poly A tail is carried out by the enzyme poly A polymerase. We know that in every mRNA, in case of eukaryotic mRNA, there is a addition of a poly A tail in the 3' end, and there is a capping in the 5' end. So poly A polymerase is a template independent enzyme. Yes, poly A polymerase is a template independent enzyme. So and another template independent enzyme is TDT, or terminal deoxynucleotidal transferase. So two template independent enzymes. Here poly A polymerase is a template independent enzymes. Number B is the polypolymerase catalyze the addition of AMP from ATP to the 3' end of mRNA. It's not correct. Polypolymerase is option C. Polypolymerase is a RNA template dependent enzyme. It's not correct. It's a template independent enzyme. So D, polypolymerase catalyze the addition of ADP from ATP to the 3' end of the mRNA. No, polypolymerase catalyze the addition of AMP from ATP to the 3' end of the mRNA. Polypolymerase add AMP to the 3' end of the mRNA. So these are option E. This polypolymerase catalyze the addition of AMP from ATP to the 3' end of the mRNA. These are correct. And polypolymerase is a template independent enzyme. This is also correct. Polypolymerase catalyze the addition of AMP from ADP. No, this is also not correct. So these are answers. Option 3, that is A and E, is the correct answer. So polypolymerase is a template independent enzyme. So next question: RNA editing, a post-transcriptional process, is achieved with the help of guide RNA. Which one of the following statement about the process is not true? We know that RNA editing occurs by two processes there in RNA editing. One is addition of U residue, another is the deamination of cytosine to uracil. So RNA editing occurs via two processes are there. One is addition of U residue, addition of U residue, addition or deletion of U residue, of U residue. Another is the modification of deamination of cytosine to uracil, deamination of cytosine converted to uracil. Deamination of cytosine is converted to uracil. These are all are mediated by guide RNA or gRNA mediated. Now here you have to see that not true. So gRNA dependent RNA editing happens in kinetoplast DNA. Yes, more in gRNA dependent RNA editing occurs in kinetoplast DNA as well as kinetoplast mitochondria. And gRNA involved in chemical modification of tRNA? No, gRNA is not involved in chemical modification of tRNA. C, option 3 is this process involve insertion or deletion of uridines. Yes, they are involved in addition or deletion of U residue. The sequence edited ones may be re-edited using a second guide RNA. Yes, these are also correct. So option 2 is the wrong answer. That is the guide RNA insertion. It's a chemical modification to of tRNA. So the mechanism of U insertion and deletion, RNA editing in kinetoplast mitochondria. Now, next question. With the intention to identify the genes expressed in an organism at specific stage of development, mRNA were isolated from given organism. Now, see that question actually. Now here actually RNA editing. Now see the question is saying that there is a particular gene is there. This is a gene. Now gene, then it produces mRNA, and mRNA is converted to cDNA. mRNA is converted into cDNA. Okay. mRNA is converted to cDNA. Now these are gene, and this gene, the mRNA converted to cDNA. Now cDNA do not have any match with the mRNA, so there is a cDNA was synthesized and cloned in a suitable vectors, and a few of the cDNA sequence showed no matches with the genomic DNA. There is no sequence match, almost no sequence match with the genomic DNA sequence, and further it was observed that sequence where U reached and found to be stretched dispersed along the sequence, and all it is found that the sequence at U-reach sequence, we know that there is a possibility is RNA editing is occur. Because of RNA editing, there is a post-transcriptional modification, and it's a deamination of cytosine to uracil. Another is the guide RNA mediated insertion and deletion of U residues is the possible. Because in case of splicing, if we see in case of splicing, in splicing process, if we see during splicing process, during normal splicing, during normal splicing, just introns are cut out and exons are joined together. Introns are cut out and exons are joined together to form the, suppose these have a intron and introns 1, intron, these are exon, intron, exon 2, and exon, intron, this is also intron. Now these introns will be removed, these are exon 3. Now this E1 will join E1, E2 and E3. These three will join together, and intron will remove from here, and these is form the mature mRNA. So mature mRNA comprises of only exons. These are normal splicing. But in case of what happened in case of trans splicing or alternate splicing, during alternate splicing, the several these are like E1 during alternate splicing, E1 and E3 form one copy, and E2 and E3 are joined together, and they form like the exon 2 and exon 3. Here exon 1 is absent, so two different protein product will be formed. These are alternating splicing. But in case of trans splicing in several RNA are joined together. During trans splicing, what happened that suppose these are from one gene, and another, these are from one mRNA and also from one gene product, gene 1 and gene 2, another, suppose EA and EB, exon AEA and exon B, and here E1, E2 and E3. Now these two separate gene E2 combined with EA, this form in mature mRNA E2 or EA, then it's a trans-splicing, because trans-splicing are the only possibilities is that the following only possibility: the guide RNA mediated insertion of U residues involving terminal transferase and RNA ligase. So these are, D is the correct here, and deamination of convert to cytosine or uracil, C or 2U. So D and E are both are possible. Now next question. The leader sequence in some of the protozoan parasite is transcribed elsewhere in the parasite genome at gate joined together with several transcripts that make up the functional mRNA. Right, that leader sequence of two protozoan parasite is transcribed elsewhere in the parasite genome and get joined with the several transcripts that make up the functional mRNA. So this is the joining of the transcript occurs by the process of is a trans-splicing. This occurs due to the presence of trans-splicing. This happened due to trans-splicing. So answer is option 2 is the correct answer. So option 2 is the trans-splicing is the correct answer. So next question. So a gene gives two different products in two tissues. No epigenetic modification in the gene could be detected, and in the mRNA no addition of U's were found. This could be due to a gene have two different product in two tissues. Suppose gene A produce one product in brain and another product in the liver. Now there is no epigenetic modification like methylation. There is no epigenetic modification could be detected, and modification is detected, and mRNA and no addition of U's are found. That means here deamination is not possible. Deamination, so deamination is not possible here. One of the important is deamination is not possible here. Second is that alternative splicing is all alternative splicing. We know that different product can be possible from alternative splicing from one mRNA transcript without any epigenetic modification. So mutation, no trans-splicing, no. So answer is alternative splicing. Alternative splicing can be produced. Suppose these genes are there, E1, exon 1, exon 2 and exon 3 and exon 4, and now these E1 and E4 joined produce one mRNA product, and E2 and E3 produce another. These are produced in the brain and these are produced in the liver, suppose, and this can be produced. So two different gene product or two different protein can be possible from one single mRNA. So these are due to alternative splicing. So next question. Now, which of the following process does not take place in 5' to 3' direction? We know that 5' to 3 most of the like DNA replication, transcription and nick translation also occurs in 5' to 3' direction. Only the RNA editing does not occur in 5' to 3' direction. So answer is option 4 is the correct answer. So next question.